So we continue with muscle physiology. We're going to talk about first muscle tension. So as the sliding filament theory works, as calcium is released and binding onto the troponin, cross bridges are being made. But as more calcium gets released, more cross bridges are made, so there is a stronger and stronger and stronger contraction until all of the troponin molecules have calcium on it. And then as the calcium starts to get pumped back into the sarcoplasmic reticulum, there will be fewer and fewer and fewer being able to make cross bridges. So it's not an all or nothing thing. It ramps up and then it ramps down. So when we look at the graph of it, it looks like this. So there is the muscle is relaxed, and right here is when the acetylcholine is released into the synaptic cleft. And from here to here is all the excitation events happening. So this is when the depolarization of the sarcolemma happens. This is the action potential carrying on. This is the depolarization of the sarcoplasmic reticulum. This is the release of the calcium right there. And then a little bit of calcium binds onto troponin, and tropomyosin is moved out of the way, and we have some contraction, some cross bridging, and there's more and more and more as more calcium is released, and more calcium is released, and then right about here, the calcium channels close, and the pumps start taking over, and the calcium goes down, and so does the number of cross bridges happening. So the power of the contraction goes back down to resting. This is called a single muscle twitch. The period between the stimulus, the release of the acetylcholine at the synapse with the motor neuron, and the time of the first cross bridge is called the latent period. This is the time after the trigger is pulled before the bullet exits the gun. It's going to happen. There's no stopping it in here. It's just hasn't resulted in a contraction yet. There is this maximum, but there's this steepness of curve up and down in the contraction period and in the relaxation period. The steeper these are, the faster the twitch. The more spread out it is, the slower the twitch. So the latent period, and these periods are important, so please know them. It's the minimum. So it's the time between the stimulus but before contraction. So there's no contraction happens during it. Then we have the time of building contraction and then the relaxation. Okay. If the muscle is stimulated repeatedly, what starts to happen is more than one motor unit starts to get the signal and starts to contract. At first, there's very few motor units, and then more and more get called in until we can increase the force enough to move the load. So muscle can, the same muscles that can caress your face also can slap your face, depending on the recruitment and the of motor units and the summation. Right? The way that we recruit motor units is based on the size of the motor units. Small ones are the first to be stimulated, the large ones are the last. And we build up with pressure until we can accomplish what we're planning to accomplish. Okay? So motor unit 1 gives you this much tension, add 2, and you get that much, add 3, and you get this much, and you have stronger and stronger contractions. I'm going to tell a true story of what happened with me one time, and I think about this often when I do this topic. A friend of mine, a very close friend of mine, who was a professor at Georgian for 35 years, uh, but we were friends from way back in university days in Hamilton. Uh, he had a master's degree in English literature, and so you can imagine how many books the guy had. Huge number of books. And one time we were moving him. All his friends were helping him move, actually to Barrie from Peterborough. And he had packed his entire apartment into boxes, and he went out and he bought 2 cubic foot and 4 cubic foot boxes uh, from a moving company. So all the boxes looked the exact same, and he had packed everything. Well, in his library, he had a nice chair for reading, and he had a desk, and he had a couple of thousand books. And so he had packed all of these books in these boxes, and they were heavy, heavy, heavy boxes. And go into the room, pick up this heavy book of boxes and carry it up, put it in the truck, and then go back and get another heavy book of boxes and put it in the truck. There were lots of boxes of books, and they were really heavy. What I didn't realize is that he had some cushions on his chair, and he put those in a box. It looked the exact same as all the rest of the boxes. So after about moving 10 or 12 boxes of books, I went in and I picked up this box of pillows. And I ended up throwing the pillows into the air because I had recruited so many motor units to lift this heavy box of books that my expectation was that the next box was going to be equally as heavy. And so you recruit and use all those motor units right off the bat. And it was way too many for the task when the box was filled with pillows. And that is indicative of how these motor unit summations work. So that is kind of that's a summation of numbers. We can also have something called wave summation, which is a temporal summation. And so if the twitch runs its whole course, there's quite a bit of time, the latent period, the period of contraction, and the period of relaxation. But what happens if the next stimulus happens before the period of relaxation is even started, let alone completed? Then the contractile force adds up and you get a higher tension, right? And so the muscle fiber never truly relaxes because the next stimulus has started the next wave of contraction before the previous part of relaxation has completed. And so the amount of force goes down in the relaxation and then back up again for the next contraction. And so when the muscle never goes all the way back to relaxed, we call this tetany. And there's 2 types of tetany. There's incomplete tetany and there is full tetany. Uh, the problem with this is that it leads to fatigue, that the contraction is using up ATP so quickly and producing probably pH changes and a few other things that the muscle will fatigue. This is part of your physiology assignment, so I'll let you kind of deal with that, and so I'm not going to beat it right now. But if this is a muscle twitch, and the muscle completely relaxes, and then the next stimulus happens, and we get the latent period and contraction relaxation, latent period contraction relaxation. We get this, and these are twitches. If the if the it comes quicker, then we end up with this next contraction happening before relaxation in the next one, and it builds here until all of the troponin has uh, calcium, and we have the maximum number of cross bridges. So we're releasing calcium before the calcium is pumped away. So now there's even more calcium and more calcium until we get here. We call this the incomplete tetany, that it goes back and forth. But as the frequency goes up, we hit this point where the next stimulus happens during the previous twitches period of contraction. And so it just flattens out at the max. And then it drops off with fatigue. We call this wave summation. Now, the muscle is never completely at rest. Different motor units are completely at rest, but there's usually a couple of motor units in a muscle contracting at any given time. So there's a resting tension in the muscle. It's involuntary. It's just random stimulations of motor units. And it's always different motor units, so there's no fatigue involved. There's no movement happens here. It's just muscle tone, as opposed to complete flaccid muscle. And if you've ever seen a neurological impairment that leads to flaccid paralysis, you would definitely understand what tone is. Okay. Contraction is really about moving making the sarcomere shorter, moving the Z disc closer together, shortening the length of the muscle fiber. But it's not. It's tension in the direction of shortening the muscle fiber. So we can have isometric and isotonic contractions. So an isometric contraction is when the cross bridges happen and there's tension being created by the cross bridges, but the thin filaments are not moving because the load that you're trying to move is greater than the tension that the muscle is able to produce. So isometric, same length. The muscle doesn't change in length during this. So this is holding a weight and not having your arm move. It's that kind of thing. Isotonic contractions are muscle tension overcomes the resistance, the load, and movement happens. And once the movement happens, the tension levels off. So you don't create more tension, uh, generally speaking. You can if you want to accelerate something, but generally speaking, once you got the object moving, that's the amount of tone that you use, the amount of tension that you have. So it's isotonic. The tension stays the same, but the length changes. Concentric is when the muscle, in fact, shortens, and we use this for like if you're doing a curl with your biceps, the muscle gets shorter, the weight moves towards you. Um, that's a concentric contraction, a concentric isotonic contraction. But you can also have eccentric contractions. You use this every time you sit down. Uh, you go from standing to sitting. Your gluteal muscles uh, are fully contracted in standing. They're relaxed and stretched in sitting, but the controlled act of sitting is progressively lessening the tension so that the muscle, in fact, gets longer with control. This is how you gently put down an object that you've just picked up with concentric. You eccentrically can put it down. When you pick up your beer off the table and bring it to your lips, that's a concentric contraction. If you're holding it to say cheers, that's an isometric contraction. After you've taken a sip and you put it back down on the table, that is an eccentric contraction. And that's just illustrative of it. So almost all contractions will have an isometric phase. So this is the time of the period of contraction when the muscle tension hasn't built up enough to uh, to move the load. So as you reach for the glass and you start to pick it up, but you haven't moved it off the table yet, that's the isometric phase. Then you have the concentric as you're getting the movement and the muscle shortens. Right? So the difference between these two graphs is that this red line is muscle tension and this red line is muscle length. You'll notice the muscle length stays the same, muscle tension changes in isometric. You hit this point and the muscle tension flattens out, right? But the length goes down. That would be a isotonic concentric contraction. If the muscle length goes up while muscle tension stays the same, it's isotonic. You have to be careful about some of these things. So one of the big problems with isometric is that you see blood pressure go up. You'll see that in people that are holding a heavy weight for a long period of time, their face starts to turn red. And so this can be a concern for people with pathologies. That's all that this is saying. Okay, there's a relationship between the length of the muscle and the tension that it can produce. So if the thin and thick filaments overlap a certain amount at rest, the resting length, you're going to get maximum amount of contractile force because it's the optimal overlap of the thick and thin filaments, so that when the cross bridging happens, there are lots of myosin heads that can cross bridge to the thin filament actin molecules. If the muscle fiber is short, there's already quite a bit of overlap. And so what's going to happen is the thin filament, half the thin filament is already going to be past the myosin heads. And there's really no place for the thin filaments to go. They're already most of the way to the midline. And so the movement is limited, and so you get weaker force when the muscle is short. When the muscle is stretched and extended, then what happens is the thin and thick filaments don't overlap enough for a lot of cross bridges to happen. So we get a weak force at the extended length as well. Right? So, so what's going to happen is a chart that looks like this, that if it's contracted and it's already overlapped, we're going to have very little tension. When it gets to its resting length, we're going to have the maximum. And then as it stretches and we have less and less overlap, it goes right down. If the muscle is pulled, it's at the point where there's no more overlap and no tension can happen. So the sarcomere length is, for maximum contraction, is from about 75% of sarcomere length to about there. So if the sarcomere length at rest is 2, it's from about 1.5 to about 2.5 length. So from some authors will say from 50% under to 50% over it, but it's going to max. Like there's kind of a peak here. So one of the things that's interesting then is that if you stretch before you exercise, you will create the maximum amount of tension, because you'll start off here and then you will hit here at your stride. Be able to lift more weight if you stretch first, but not too much. It's got to be just pulling it out. Most of us are chronically contracted because of tension and blood pressure and stress and things like that. So stretching is a very important thing. Okay, muscle fatigue is a physiological inability to produce tension. Right? There's a lot of different theories as what causes muscle fatigue. We don't entirely know. We have some very good guesses. We have some very good candidates, but it's a very complex thing. This is not tired. This is not a psychological inability to go on. This is a physiological inability to make cross bridges happen. So there's a couple of things will happen. We'll use up the glycogen stores in prolonged exercise. And this is especially for the fast glycolytic fibers that have glycogen stored in it. So those muscles fatigue quickly because you'll use up the glycogen. There's nothing else to oxidize to make ATP. Another real problem is that you're using up the calcium at the motor neuron, and so you're not releasing as much acetylcholine into the synaptic cleft. And further, if you've been using it a lot, there may not be enough acetylcholine vesicles at the motor neuron left, and because the motor neuron has to replenish that stuff, and it takes rest to replenish. So there's a problem with excitation at the neuromuscular junction with not releasing enough acetylcholine, either because not enough calcium or not enough acetylcholine, or both. Right. Another problem is if there's too much calcium released from the sarcoplasmic reticulum, it's in the sarcoplasm, and calcium is 2 plus. It's a strong cation. This alters the charge within the muscle fiber itself and makes the depolarization, the whole action potential, not work properly. Because if you remember, the action potential depends on there being a concentration gradient caused by the sodium and the potassium, with the outside being more positive. Well, if the inside is too positive, then that's a problem. So you don't get the action potentials the same way, right? If there's too much P from the ATP in the cytoplasm, in the sarcoplasm, then there's a concentration gradient that slows the release of the P from the ATP, ADP, and so it doesn't work. And the P which is a negative, will bond to the calcium and make less calcium available for troponin. So that's a problem too. And there's numerous other problems that you're going to encounter. So the next thing we're going to talk about is what happens when you exercise. So there's two types of exercise. There's endurance exercise and there's resistance exercise. The endurance exercise is what it sounds like. So this is lots of reps, low weight, for lack of a better description. So these are the things that require endurance. And so endurance requires more ATP, and more ATP really means more mitochondria, more myoglobin. We get endurance mostly from the slow oxidative fibers. And the more you use these slow oxidative fibers, the better their ATP production. We will grow more blood vessels to supply oxygen and nutrients. We will grow more mitochondria within the cell to keep you in ATP for a longer period of time. There will be more oxygen storage and so the meat will get redder because there's more myoglobin. And that improves with endurance training. Resistance exercise is high weight, low reps, so lots of resistance. And this leads to hypertrophy and of the muscle. It doesn't lead to hyperplasia. You don't get more muscle fibers. You get increased size of each muscle fiber. And that's really because what you're going to grow is more myofibrils, more sarcomeres. And so, each fiber will get bigger, especially the fast glycolytic fibers, because they're the ones that are able to produce the most power. They're the largest fibers. Think of white meat. You increase the glycogen storage in them. You will also increase mitochondria to a certain extent, but nowhere near the the increase in glycogen and things like that. Um, Oxygen delivery doesn't matter all that much when you're resistance. So that's why if you want to bulk up, you use resistance. And if you want to tone up, you use endurance. Right? Now, we can have a very limited amount of hyperplasia, the increase in number of muscle fibers, but it is very limited. If you don't use muscle, if there's a lack of exercise, you end up with atrophy. And atrophy is the opposite of hypertrophy. It's a decrease in size due to lack of use. Uh, you in It's usually reversible. If you've ever had a cast, you will know what atrophy looks like. You'll see it in people that end up in wheelchairs and things like that. There'll be atrophy of the calf muscles, etc. Um, if it's really extreme, it becomes irreversible. The atrophy gets to the point where the muscle fibers die, and then they can't be brought back. As we age, we lose muscle mass, and it happens pretty early, usually in the mid 30s. You can postpone that a lot by going to the gym, by working out. So we lose size, power, and endurance of muscles. The fibers get smaller in diameter. They lose myofibrils. We lose the ability to store oxygen. Sometimes impaired circulatory supply uh, happens, and we end up with fibrosis, connective tissue, scar tissue within the muscles, especially from old injuries and things like that. And these things are cumulative. And as we age, all of these things add up. And unfortunately, it's kind of self fulfilling because as you lose power and endurance, you exercise less, which then causes a loss in diameter, which then causes a loss in oxygen delivering capability, et cetera, et cetera, et cetera. And this ramps up. The speed at which these changes happen really are lifestyle dependent, but inevitably it goes a certain way. I'm nowhere near as strong as I was 25 or 30 years ago. Uh, and I know it. And it takes me longer to recover uh, from any damage than it ever used to. We're not going to talk about steroids or any of these things. Uh, the cardiac muscle, we're going to talk about when we do the cardiovascular system. So I'm going to skip over all of these. Smooth muscle is in the walls of hollow organs, and it's a different thing. And again, I'm just going to very quickly look at this. So we have it in our blood vessels, in the intestines, in the bronchioles, in the ureters, um, in reproductive system tubes, etc., all over the body. Right? It almost looks like a stratified epithelium. The cells are fusiform, which means they look like it looks like Bob Marley maybe rolled them at 420. Uh, they are small little cells. Uh, they have a sarcolemma, but the calcium channels are through the sarcolemma. There's no transverse tubules because they're small. They have the surface is got little caves in them called calveoli. And we don't store calcium within the cell. It goes to the exterior into the interstitium and comes in to cause contraction. So the cells look like this, and this kind of net over top of them is, instead of the thin filaments being attached to a Z disc, they're attached to proteins on the surface. And so the thin and thick filaments are all kind of arranged like a net around the surface of the cell. So they're anchored there. There's lots of intermediate filaments, and we call the points where they interact as dense bodies. So the contractile proteins, which are still actin and myosin, kind of join onto that. There's no sarcomeres, there's no Z disc, so there's no striations. So there's actin and myosin, and that's basically it. So the myosin heads are along the entire length. So there's lots of additional cross bridges can happen compared to skeletal muscle. The way that the myosin attaches to the actin, we can utilize that by not using ATP to disconnect it. So if we need the muscle to stay contracted for an extended period of time, it just latches on and stays there. It's almost like rigor. And we don't use up ATP to maintain this. And when you consider that your anal sphincter is smooth muscle, this is a good thing. You don't have to use ATP to keep it closed. Instead of the calcium binding to troponin, it binds to something called calmodulin, and that triggers the contraction. So it's not the same sort of pull the rope out of the way sort of thing. So the calcium comes in, the calcium interacts, activates some enzymes in the calmodulin chain that cause the cross bridge to happen. And we'll just leave it at that. We don't have to go into too much detail. So when the stimulus opens voltage-gated calcium channels, there's no action potential. The calcium channels open. Calcium enters and binds to the calmodulin. The calmodulin forms a complex with some enzymes, kinases, and then we end up, the kinase then works on the myosin head, and the cross bridge forms. And as the thin filaments slide towards the center of the thick filament, it pulls on the dense bodies, and because the dense bodies are all joined together by intermediate filaments, the whole cell gets shorter with tension. You take away the stimulation, calcium pumps remove calcium from the sarcoplasm, and then the myosin takes time to dephosphorylate, and then so we end up with this latch bridge. We end up with temporary rigor, and so once it's contracted, it's very slow to release, and can stay contracted without using very much ATP. There's a huge long latent period. It's very slow ATPase. There's the contraction is long in duration because it takes a long time to pump the calcium out. We have to dephosphorylize the myosin head, and while it's dephosphorylizing, we have the cross bridge stays latched. We don't need this to happen quickly because we're not using this for purposeful motion. We're using it for peristalsis. We're using it for motion along the intestines, for blood pressure, things like that. Completely fatigue resistant. I mean, you can maintain with using very little ATP. The length and tension is huge. From twice its length to half its length is the tension curve, as opposed to 50% above and 50% below. It's involuntary. So it depends on signals from the autonomic nervous system. It's interesting, smooth muscle will have different receptors for different types of neurotransmitters. So, for example, smooth muscles in the bronchioles contract in response to acetylcholine and relax in response to norepinephrine, where in blood vessels, you're going to have a contraction in response to norepinephrine and a relaxation to acetylcholine. Right? Smooth muscle will automatically contract in response to being stretched. This is why when feces move down into your rectum, you have the urgency to go. And you have to use the skeletal muscle to hold the sphincter closed in order to get to the bathroom. It's the stretch of the walls of the rectum causes the rectum to contract and increase the pressure. Right? But here's the problem: is that the smooth muscle can adapt to that very quickly. So this explains why you have to go, and you're holding it. And then by the time you get to the gas station, you don't have to go anymore because we've had this stress relaxation response. Smooth muscle will change depending on pH and oxygen levels and certain drugs and all kinds of things like that. There's two types of smooth muscle. So there's multi-unit smooth muscle, and so these are cells that act independently. There's multiple units, and so we use this like in the iris of the eyes and in the rector pilae of the skin, in passageways in the respiratory system to deliver oxygen to certain areas. They react rather similar to skeletal muscle. Each group of smooth muscle cells, or down to a single cell, acts on its own as a single, as a unit. And so given a handful of smooth muscle cells, we have multiple units, each one behaving independently. The vast majority of smooth muscle is single unit, and that's where all of the muscle cells, all the smooth muscle cells, contract together as a single unit. So these are the walls of your digestive system, your urinary tract, your bladder, in the walls of your blood vessels, etc. Most of it is single unit. The autonomic neurons, there's no synapse. There's just swellings along the autonomic neurons that release neurotransmitters, either acetylcholine or norepinephrine. Acetylcholine if it's the parasympathetics, norepinephrine if it's the sympathetics. And the neurotransmitter just goes right across the whole sheet of cells. And so there's no specific junction. It's really diffuse. So all kinds of cells get stimulated simultaneously. And there's gap junctions between cells. So when one depolarizes and starts to contract, it causes its neighbors to polarize and contract. So here are the varicosities, and they just release just a flood of neurotransmitter over the entire area. So that's it for muscle physiology.